thank you so much for being here. I can't tell you how excited I am to have actual real people in the room and actually have an actual function with people outside. It's one of the first events we've had in the school, and it gives me great pleasure to host you guys here tonight. And I'd welcome all the friends of the school and the alumni of the school, our staff and people across all different sectors of veterinary science, which is amazing. So my name is Jacqueline Norris. I'm the incoming head of school and dean for the vet school, starting next week. So I've got in a little bit early. I hope that after this event you will join us for a drink and a conversation, things that we've missed during the dreaded COVID. So obviously tonight we're here for the Ian Beveridge Memorial Lecture, and Ian Beveridge was very much known for his pioneering of what was called One Medicine then. I guess we call it more One Health now, which our wonderful speaker will outline more. But obviously I want to start by acknowledging the country on which we are here at the moment. We're on the land of the Gadigal people of the Eora Nation. And if we think of the ancestral lands on the University of Sydney and all of our country across Australia, the Gadigal people and other people really embodied One Health. They took care of the land, they took care of animals and people and environment in exactly the way as we think of with One Health. Their traditional knowledge of our land, its plants, animals, they lived and embodied the true concepts of One Health. Evidence of that is in their custodianship of our land and our landscapes. The animals and fish and plants that are etched on our rocks that you'll see across Sydney and many parts of Australia. And so today our school and university also strive to understand and care for our environment, care for the animals under our care, and also humans in contact with them. So I'm very proud that our school does a lot of work within this area of One Health, and it's an enormous privilege to be the incoming dean. The painting on what is for you the right-hand side there is called Ancestors, and it's a painting by Sarita King. She's a young up-and-coming artist, Indigenous artist, and I think I've always loved this painting simply for its background, where you think the interconnectivity between current and ancestors is very true for the kind of research that we're doing. So who was Professor Ian Beveridge? He was a graduate of this veterinary school. It was then a Faculty of Veterinary Science in 1931. And he was actually from a farming background. He was born in 1908 and from Junee, not very far down the south coast of New South Wales. And it was always intended, I think, he was going to be a farmer, but he had other ambitions. After graduating from vet school, he actually worked for the CSIRO in the McMaster building over one of the buildings just adjacent. And he worked on finding solutions for sheath rot in sheep and foot rot in sheep, two very, very important diseases that he discovered that just changing the alkaline of urine helped sheep with sheath rot. That's a very hard word to say several times, I tell you. And with foot rot, that's actually been a tradition in our vet school. We've been doing research and advances in foot rot, even to the discovery of autogenous vaccines. He then took a Commonwealth Fellowship and went overseas to the USA, where he worked on influenza viruses, and he discovered that swine influenza virus was very, very closely related, almost identical to the influenza virus that caused the pandemic in the 1980s, the so-called Spanish flu. Other things that he discovered, he worked on the nomenclature and classification of cancers in domestic animals, taking over an enormous number of journal articles in an entire year of a particular journal. And he established the Cambridge Veterinary School and was also the chairman for the World Veterinary Association for 18 years. So an amazing person. One of his biggest publications was the art of scientific investigation. So this tradition of having a speaker who speaks to the concept of One Health has been a very important part of our school for many years. And our speaker tonight is Dr. Barbara Brito Rodriguez. And she's a veterinarian from Chile and has originally worked, did a PhD in, sorry, I've completely lost my train of thought. Sorry, she did a Master's in Veterinary Medicine program, followed by a PhD in Epidemiology in UC Davis, which is one of the best vet schools around. Her work has focused on disease modelling and molecular epidemiology of foot and mouth disease, and you'll see that that's actually foot and mouth lesions from the 2001 outbreak in the UK. And you would have seen on the news not so long ago how important foot and mouth disease, how close it is to our borders and how devastating it would be if it got here. So Barbara also joined the UTS. She got a fellowship there where she worked for a while and developed a collaboration with EMAI and has now moved to EMAI, where she continues her work looking at the role of metagenomics and understanding how infectious diseases develop. So it's my great pleasure to introduce Dr. Barbara Rodriguez to the podium. Thank you.
Thank you very much for the introduction, and thank you very much for the invitation. It's an honor to be here. My talk today is about advances in One Health and how microbial genomics or the genes of the microbes have helped us reveal some of their many secrets. I did my PhD at UC Davis, and it was on foot and mouth disease virus. So this is a very devastating virus, not because it kills the animals. The animals mostly recover 2 weeks after they get infected. But what happens is when it's introduced in a disease-free country, the way we deal with this virus is that we have to eliminate or cull every animal that has been exposed. And these are animals that are healthy, and this can be millions of animals. This is the fastest way to get rid of this virus from a disease-free country. And people that have lived through these epidemics, they remember this because it not only have impacts in animal health, but it has impacts on human health and social and an economic impact to the country. So this is a very important disease, and it has been studied for many years. When I started my PhD, we started doing disease modeling, but we incorporated the genetic data of the virus. And the genetic data of the virus, it allows you to tell where's the virus moving. It allows you to tell which type of variants are present in different countries. So we get data from endemic country and how the virus moves. And we saw some of the lineage, we call it then lineage turnover. So there's a dominance of certain viruses, and then there comes another dominant virus that takes over that. And I don't know if this sounds familiar with COVID, in which variants take over and they dominate. And we know this about foot and mouth disease for many years. The other reason why it's important to look at the genome of virus, of foot and mouth disease virus, is because there's a vaccine that's very effective. But it only works when you have the right vaccine matching to it. So the reason I started this with foot and mouth disease is not because I'm going to do my talk on foot and mouth disease, but it's how I started my journey in microbial genomics and how I started using this tool to study other diseases. And also how foot and mouth disease may or may not fall in this concept of One Health. So One Health, this is what the WHO has as its definition, and over time I've seen, and probably you've seen, many different definitions of the One Health concept. But it's based in that the way in which we should approach disease is an integrated, unifying approach. And it's based that humans, domestic and wild animals, plants, we're all linked, and this is an interdependent system. So there's a big One Health umbrella in which zoonotic diseases are the center. Foot and mouth disease may fall within this umbrella, but it may be somewhere in the periphery of it. So that's why I have some examples of zoonotic disease and how we have used genomic data to understand the transmission in zoonotic disease. And this is not just a few viruses. I cannot give just a few examples because there are so many. I start thinking about, many of the zoonotic diseases that you can find and start writing it. Maybe, I don't know if you find a type in there, but there's many. And this is just a short list of everything that can be transmitted, that we know can be transmitted from humans, from animals to humans and from humans to animals and in the environment. So not only directly, for examples from bat, from pigs, from cattle to humans and vice versa, but also from vectors, from mosquitoes, from ticks. So that's part of the environment. So it's just to give a little bit of background of why we are using now more microbial genomics than before, I have to give a little bit of background on the history of genomics. So the history of genomics starts maybe with the first milestone, which is the discovery of the DNA double helix, which was in the mid-50s by Rosalind Franklin, Watson, and Crick. The next milestone was probably the development of a technology that allowed us to sequence the genomes, and also of cracking the genetic code, so of understanding what the genome, what a genome encodes, and that all the information that we are is encoded in the genome and is read, and every single cell can read that instructions and make us who we are. So the first development was in the mid-1970s with Frederick Sanger, who developed a sequencing technique that allows us to get more in-depth and start sequencing small fragments to understand genetics. And in 1976, the first virus was sequenced. And this is a very small virus here. This is a bacteriophage. And this virus infects bacteria. And it has a very tiny genome. That's why they were able to complete the genome. It's very, nowadays that we can sequence high throughput to say the complete genome of a bacteriophage, it sounds funny, but at that point it was an important milestone. The next milestone was the sequencing of the first bacterium, which is Haemophilus influenzae, which at the moment and historically was an important bacterium because it was thought to be the causal agent of the Spanish flu in 1918 when people didn't know much about viruses. So it was a very popular bacterial study. Then they realized that it was also present in healthy people. And years after, only in the 1930s, they realized it was a virus. And this bacteria has a relatively tiny virus, and the genome was completed in 1995. Now, the next important milestone in genomics was the launch of a very large project, which was the Human Genome Project. And this was a commitment between governments, between universities in different countries, and also with the biotechnological companies. They started the project in 1990, and they thought they were going to complete the whole human genome in 15 years. They completed it in 11 years. And people that work in research, they know that this never happens. You never complete the project ahead of time. And this happened because the biotech companies developed a sequencing technique that allowed high throughput. So to put many sequences at the same time and to sequence millions and millions of fragments at the same time. And this was what actually made the project succeed. After the first human genome, other genomes came. For example, this is the first genome of a parasite. So this is Plasmodium falciparum, the one that causes malaria. And after that, everything came with different applications. So in 2005, the first genome of a dog was completed. In 2007, that of a cat, 2009, the boa, in 2012, pigs. And then after completing the first draft genomes, the interest was put into understanding the genetic differences between people. So started sequencing genomes from different people and understanding their differences. There's also a very big project. This is an Australian project, the 1,000 Bull Genome Project. It's Australian-based to try to understand the difference. It was launched in 2010, so now we know about the difference in the genomes, but to actually do genetic selection of animals, not only based on their phenotypes, but also based on their genetics. It's also used to study rare genetic diseases, mostly in humans and rarely when you can do the whole genome sequencing. We not only sequence DNA, but we can sequence RNA. And with sequencing the RNA, we can understand what the cell is actually expressing. The cell from our liver will be different from the cell from our skin because we are reading the genetic code different. It's the same genetic code, but we are reading it differently. And to understand what the cell is doing, we have to understand what's this product that we are using to translate and to create protein. It also boosted the study of epigenetics, so the way in which we control gene expression. And important for this talk, how it has been used, how all this genomic and these applications have been used in microbial genomic. So probably one of the most popular fields is the field of studying the microbiome. And this has completely changed our view of our relationship with bacteria. Before we thought bacteria were mainly pathogens that were causing disease, but now we know that they're an integral system with us, and our health depends on the balance with our microbiome. So disbalances in our microbiome can cause diseases, not only gut diseases in terms of the gut microbiome, but it can also cause autism. It has been associated with depression and with other diseases. So we know that it's part of us, it's part of our system. Some people call it part of our genome as well. And the application in the fields of veterinary medicine are also very wide, but for example, one important one is understanding the rumen microbiome. So we know already that cattle and ruminants, they use bacteria to digest the fiber, but when they do so, they do a lot of methane emissions. So by engineering this microbiome and by understanding the whole range of bacteria that's present there, maybe we can find more efficient ways for the cows to digest this fiber. And also the microbiome of small animals, and to understand how it affects their health, how their microbiome is related to us. Other fields in microbial genomics, for example, are to use whole genome sequencing to trace outbreaks. We can use it for viruses, we can use it for bacteria, and probably most of you have seen a lot of studies that have used this technique to understand the disease transmission and to understand disease epidemiology, so in the field of molecular epidemiology. And I'm going to talk a little bit more in depth about metagenomics in the final slides, but that's the ultimate tool that allows you to understand and to discover pathogens and to understand the whole range as well of microorganisms that are present in a sample. So I was talking to a few people before coming to coming here, and they say we're not interested in the methods, we're interested in the findings. And I was like, oh, I have some methods slides, but that's because I want you to understand the results that I'm showing you later. So I don't know if many of you have seen phylogenetic trees, probably from COVID and probably from if you've seen some research in disease transmission in outbreak investigation, you will see phylogenetic trees. But probably no one tells you how to read it, not because it's very intuitive. If two different viruses or two different bacteria are clustered together, that means they are very closely genetically related, and probably you can infer transmission from them. Phylogenetic trees were not created and were not developed to study microbes. They were developed to study evolutionary biology, to understand the relationship between species. But for two different species of birds to evolve and create these phylogenetic trees, you need thousands and thousands of years. Microbiology started realizing that this tool was particularly interesting to study microbes because they evolve really fast. And so they start diverging, and you can see this branching pattern which relates to their epidemiology, their transmission, and their evolution. So you can use these trees to infer transmission, and it will depend on what the branch length means. So normally the branch length of phylogenetic tree will mean that this is a certain genetic distance. For example, these viruses might be more genetically related than these two viruses there, although both of them are in the same branch of the tree. So that's something that you have to look at in a phylogenetic tree. The numbers there also show you the statistical support, which a lot of research papers now don't put that. So when there's a low number there, that means that the tree is not really sure if those sequences should be put together, if they're genetically related. The reason I wanted to show you this tree is because I want to contrast it to this other tree that you will see in other research. This is a time-calibrated tree. And so in this tree, not only the genetic distance between the different organisms that you have here are incorporated, but also the time component. So for example, if you have a virus that was collected in 2018, a virus that was collected in 2017 and 2016, you can infer that this is not just a genetic distance between virus A and B, but it's genetic distance in time. And you can infer which is the time to most recent common ancestor between virus A and virus B. Why is this important? Because when you're trying to infer, for example, an introduction of a virus into a new geographic area, you go back to all the reference that you have, and you want to know when the ancestor of these two viruses existed, because it can help you understand their introduction point in time. So I wanted to show you this method because this tree is a time tree, a time-calibrated tree. And this is just one of the examples that I have, and it's a very good One Health example and to show you what we have learned about swine influenza over the years. These are my colleagues in Chile. This is Victor here, the lead virologist at Universidad de Chile. This is Juan. He was a PhD student at a time. And we were trying to understand the genetic diversity of swine influenza viruses in swine farms in Chile, in South America. So we went to different farms, we sequenced the virus. So the influenza virus has 8 different segments, so 8 different genes. And normally the trees that you see is only from one of those segments, which is the hemagglutinin, which is the most variable segment. It changes more over time, so that's why people use it to trace outbreak and to infer transmission. But these viruses, when 2 different viruses co-infect a different cell, because they are segmented, they really like to reassort, to reorganize themselves and to create new viruses. And actually that's how the 2009 influenza pandemic emerged. It was because of reorganization of the segments of the virus from different species. Now from this tree, what I wanted to show you, we color-coded here the viruses that we're taking from Chilean farms, which are this green here, color-coded then other viruses from pigs in South America, from pigs from other parts of the world that were not South America. We had viruses from humans in Chile and from humans in other parts of the world. And we put this together because we wanted to understand there was a lot of viruses that were from this lineage. So this was not the only virus that was circulating among pigs. This was just one of the lineages that was circulating there. The 2001, the H1N1 to 2009 pandemic virus. So what we see here, if the viruses that were circulating in pigs had been only from one transmission event from humans into the pigs, we would see all the green tips here clustered together in one big branch of the tree. But we don't see that. We see them spread all across the tree, and we see that there's different introductions here of this virus. So and across different years here, if you look at the time to most recent common ancestor of this virus is here, there were many introductions. So what happened is that this virus was very easily transmitted from humans into pigs, and it was introduced many times into the pig farms. Why is this important? Because when we look at the other lineages that were circulating across pigs, We saw that, for example, most of the virus cluster together, and they don't have a recent common ancestor with other known viruses. So that means that these are pig-adapted viruses that have been evolving only in the pig population for many years. But when we look back in time, this virus has a common ancestor with a human seasonal influenza. And this study was replicated in many countries. They all have their own swine-adapted viruses, but most of them have their origins in one of the waves of seasonal influenza. So every once in a while, there's a seasonal influenza virus in humans that get easily transmitted into the pig and becomes endemic in the pig. And a lot of the viruses that we see in pig, they started with that reverse zoonotic transmission. This is another example. This is not my example, but I thought it was a beautiful study that exemplified how you can use genomic sequencing to understand transmission and transmission of a bacteria in livestock and wildlife. So what the authors did, this study was conducted in Kenya, so in the suburban areas of Nairobi. And what they did, they collected over 1,300 isolates of E. coli from 99 different households. And they collected samples from the humans, from the livestock that were present in those households, and from the wildlife around, so bats and rodents and other wildlife that was around. And they collected over 1,300 of isolates, and they did whole genome sequencing on them. So the first thing that you can see in this tree is that they did the sequence type, and if you're used to look at the sequence bacteria, most of the time the first things that come is the information about the sequence type, and there's different sequence type that might be adapted to a specific host, so to humans or to livestock. But what you see here is most of this sequence type, this is a human distribution. There's no grouping of the humans, there's no grouping of the livestock, and there's no grouping of the wildlife. So apparently what this makes you think is that most of this sequence type, they get transmitted between livestock and humans and wildlife. The other thing that you can see from this tree is that there's many sequence types that were published in this study that are new sequence type that are not part of the reference that are published in public databases. So when you go and do a metagenomic study or a genomic study in a developing country or an underdeveloped country, you will find a lot of new things because most of our genomic sequence is basically done in developed countries. So there was a lot of new type of sequences of E. coli. And the interesting things that the authors did is that they went and looked at the pair difference between the whole genome of the E. coli and looked at those pair of sequences that were very similar to each other. So they had only a few nucleotide difference. And when you have two whole genome sequences that have very little difference between them, you can infer that might have been a direct transmission over the few months before. So they only looked at the pairs that were very closely related, and they looked if these pairs corresponded to a pair of sequences taken from the same household or between households. And if it was between humans, for example, this is livestock birds, so this is between poultry or between the same species, basically in the same household, or between different species. What they found, and this is with most pathogens and with most microorganisms, most of the transmission is within the household and is within the same species. But every once in a while, you have an event of transmission between livestock and humans, and between livestock and wildlife, and between humans and wildlife. The microbes, I don't think that they cross the species barriers very often, even E. coli, but every once in a while they do. The other interesting stuff that they did there, they look at the resistome. So the resistome is all the collection of antimicrobial resistant genes that are present in the whole genome of the sequence that they got. And if the collection of those genes were very similar, they pair it together as well. And interestingly, they find that the birds, the poultry in the households tend to have a very similar resistome. But then within what they did between household differences of the resistome, they find that poultry also shared a lot of that resistome. When they look at those sequences, it didn't seem that it was because the bacteria was transmitting between the livestock, but because the antimicrobial resistance, the antimicrobial pressure in different places was the same. And so they all arose, they all got into the same antimicrobial resistance profiles independently because of the use probably of the same antibiotics. So this is a very beautiful study. The reference is down there. And now, finally, I wanted to show you our studies with metagenomics. So this is what I have been doing over the past few years. And what, first of all, methods, of course, what is metagenomics? So let's say you have a sample. Let's say you have a nasal swab, and it's a clinical specimen. And in that nasal swab, especially if it's from an animal, it'll have not only our cells, not only the cattle cells, but it will have bacteria, it will have viruses, it might have parasite, and it might have fungi in there. So everything is mixed in that sample. So what you do is you extract all the genetic material from that sample, you fragment it in very tiny bits, and you have millions and millions of this fragment. And in a process called library preparation, this little fragment, you add a little adapter, and you put these millions of fragments over a surface. After you amplify these millions of fragments, and then in a massive parallel sequence, you sequence all these millions at the same time. You end up with a file that have millions of different little fragments, and then the problem is that bioinformatically you have to put the puzzle back together. You have to put this, and you have to figure out which sequence belong to what, which sequence is bacteria, which sequence is virus, and so on. The good thing is that this technique is culture independent. So to study most of the bugs, you will need to do isolation and culturing of a specific bacteria or virus. So you need to do that before doing the whole genome sequencing. With that, you go directly into the clinical samples and you culture this. And also, there's no you don't need to question your sample about something that you're looking for. You don't need to test for a specific virus, but the sample with the sequencing will tell you what's in there. And you can do this with DNA, and you can do this with RNA, and that's what I've been doing an RNA, because you can get the whole genome of RNA viruses, which are known pathogens. But you can also classify the DNA virus and bacteria that are expressing genes there, and you can do all the microbiome classification that's based on that information. This is review from different studies that have been conducted in livestock to look at viruses using metagenomics, and this was in 2020, so this is already dated. What you see here is that most of the studies are in poultry and cattle. These studies are mostly from North America, from a few countries in Europe, and from Japan and China. You don't see much of these studies in livestock. There's been a lot of novel viruses discovered in pigs, some in poultry. There's very little in small ruminants, and there's some in cattle. So we did this, we did viral metagenomics in 2 feedlots from New South Wales, and we find many of these viruses. Some of them have been reported for the first time in Australia. For example, we found influenza D virus here. We found these rhinitis virus that were very abundant. But most importantly, we found this virus that have been discovered earlier. This is called an nidovirus, and we find it in very high abundance and potentially associated with disease. This virus has never been isolated, so it has never been studied, and we don't know what's its role in disease. Until we isolate it, we will not know. But apparently, looking at healthy cases and sick cases, this virus might be associated with the disease process. We also discovered a novel virus, which is a paramyxovirus. It has also not been isolated, but it might have been more an incidental finding and not associated with disease. And this is another study that I want to show you, and it's about calf scar. We went to 2 different farms, dairy farms in New South Wales, and we collected rectal swabs from calves. So we collected 30 samples from each of the farms, so this was more of a pilot study, and we did the whole transcriptome in them. We had to pull together to create those libraries and this taxonomic profile. So this is the way we pull it. We have 4 different libraries from farm 1. And we know that the profile of healthy animals tend to be very similar. That's why we pull it. This profile also tend to be we pull together healthy animals. And we had different libraries prepared for animals that have scars. These were 2 newborns, 7 and 8 days old, that were right next to each other and had the same disease process. That's why we pulled them together. And similar to this, they were very similar in their disease. I put here a note. Some of them had respiratory disease. We didn't know it was going to impact anything that we saw in the rectal swabs, but it might have impacted that. So first, in a very general overview of what we saw here, what was classified as bacteria, as viruses, parasites, and host. We see that in most of the healthy animal, most of it is bacteria. We see that in the 3 different scars cases, we see very different profiles. One of them is mostly bacteria. One of them is mostly RNA virus, and one of them is kind of a parasite virus and bacteria here. And this parasite keeps showing up in the healthy animals. I'm just gonna go into the details of the bacteria and the virus here. So first, the bacterial profile, and I'm learning about bacterial microbiome myself. I'm more of a going to the virology side rather than the bacterial side. But what you can see here is that most of the healthy animals have rather similar profiles of the RNA, of what we could classify from their RNA there. But when you look at the animals that has scarring, you can see that there's a shift of the species. So what this is depicting is different genus of bacteria present in the total bacteria part of the samples. So you can see that in this scarring animal, it's somehow shifted and it has more genus, more abundant than other. In this one, Campylobacter jejuni and E. coli, but this represents a very tiny bit of the RNA that we sequence here. And in this one, the only thing that seems different is this Campylobacter here, that we know it might be pathogenic and it causes bacteria. Or it could be that we sample these animals later at their disease process, and there might have been a virus earlier, and we don't see that virus anymore because we sampled them afterwards in the disease process. So that's another very challenging aspect of doing field work. We don't know when those animals were initially infected. And so the viruses. So with the healthy animals, we found a lot of astroviruses, we found bopiviruses, which are viruses that have been more recently described, very abundant. Astroviruses are known to be able to cause disease, but probably in these samples they weren't doing anything. And the viral part in these libraries was so tiny that probably was very few of them in there. We see a shift in the animals that have respiratory disease to having other type of viruses. And we see that in this farm in particularly, there's a torovirus circulating that we don't see in this farm here. And these animals were healthy. They had a lot of virus, and they had this torovirus. So, and the other thing is that these animals that had respiratory disease also had a shifted virome here. So it's interesting that maybe we're picking up something that's respiratory in the rectal swabs. So what happened with the sick animals? So these animals here, most of this is virus and most of this is torovirus. But when you try to infer causality, you say, well, so in this healthy animal, there's also a lot of torovirus. So is this virus really causing disease? I don't know. In this case, this one that has scar, we saw that there's almost no virus, so we don't know. But in these ones here, most of it is virus and most of it is rotavirus. So we might be able to think that this is probably, that this is very likely a diarrhea caused by rotavirus, which is nothing new. This is probably the most common virus that affect cattle. But then, because this is a One Health talk, and the next slide is about it, is rotavirus zoonotic? Do any of them? So, so does it transmit to humans? So as in most viruses, often not. Often they are species specific, but in some cases, and same thing that we saw with other viruses, same that we saw with E. coli, in some cases it can transmit to human and it can cause zoonosis. And that's why we need to keep our genomic surveillance to understand the ones that do cause disease in humans and the ones that don't. There's vaccine available for human, for cattle, for companion animals, so it's also important to understand if the vaccination is working or not. We had a study with some colleagues at the University of Minnesota where they were doing metagenomic studies in bovine, and they found new genotypes. So the more we keep doing these metagenomic studies, we'll find more diversity of this virus, and probably this will or will not, some of them might cross the species barrier. In the case of the viruses that we found in this cow, this was very interesting because it was a co-infection. It was not just one genotype of rotavirus, it was two genotypes of rotavirus. Both of them are specific to bovine, so we were wearing gloves anyway when we took the samples, but just in case. And the other thing that's interesting with metagenomics is that we will start seeing these viruses in places where we don't expect them to be. Rotavirus, you normally expect them to be in the gastrointestinal tract. And this is another recent publication using metagenomics, and Ben House is a person that has been doing metagenomics to understand disease in livestock for many years. So what they saw, they saw pulmonary infections with porcine rotavirus A, and it was not just taking a sample, doing metagenomics and finding the virus. They actually did the whole research to understand if the virus was actually infecting the lungs, and it was infecting the lungs. So the more we do these untargeted approaches, we will start seeing viruses in places we don't expect them to be. So what are future directions for us? We have this collaborative project called Dairy App. It's a big initiative with the Dairy Research Foundation in Sydney and with the industry, so with Dairy Australia. So we plan to sample many dairy farms to understand what's circulating in these farms in a future study. And as a final remarks, this came on to my Twitter feed at the beginning of the epidemics. And this is the epidemiological triad. Everyone that study epidemiology saw this triangle many times, but they never see it with this big politics here. And so the way infectious diseases move around, it really depends not only the host, and this also is a One Health, so the host is the animal or the humans, the agent, the environment, but the legislation about it. This was when different countries were adopting different measures to control SARS-CoV-2. Some countries were mandating masking. Some countries were having more stronger policies about vaccination. And that really shaped the distribution and the dynamic of the landscape of SARS-CoV-2. So this is really important. And maybe now that Australia is planning to create a CDC, a Center for Disease Control, it's important that they have the One Health concept at the very core of their foundation and their creation. With that, I want to finish and thank all the people that have supported me during all these years, where the only thing I wanted to do was sequence everything, and I've had a lot of support from my supervisor, from Jim, from the people from the virology team at EMAI, from people at Sydney Uni, Yanni Garcia, who's the lead of the Dairy, Neri Holmes, who helped me at the beginning doing all the sequencing of how to get into the metagenomics world. The people from Skype, so this is a big consultancy, very active in dairy research, and all the team at UTS, and my collaborator in Chile, Dr. Victor Nera. And thank you very much. With this, take any questions.
Hi, a great talk. I'm not into farming much or anything, so I don't know if this really applies. And as you said, there was research from 2020 that's already out of date, which must be frustrating. But we heard that certain viruses during the last couple of years in the human sphere of things became extinct just because of the various international things that went on. And I was wondering if any of that managed to show itself happening in the agricultural world. Was there a similar sort of effect where some viruses or pathogens became extinct because of the lack of movement?
That is a very interesting question, because people stopped moving between farms and probably that impacted. I don't know with this pandemic, but I remember in the US when I was working there and in the University of Minnesota, they had a lot of a viral infection that's called Pierce virus in pigs. It's a very bad disease in pigs, and you don't have it in Australia, thankfully. But in the 2009 pandemic outbreak of swine influenza, they completely stopped pig movement. And then there was another pandemic of PED, porcine epidemic disease. And they also stopped complete movement of animals. And they could see a reduction of peers in those years that they weren't expecting. And it was because, of course, they stopped the animal movement there. So I wouldn't be surprised if that was also the case for COVID.
Thank you, Barbara. That was a fantastic talk. Something that's always, as an epidemiologist, something that's always troubled me with phylogenetics, genomics, is this conclusion that if two organisms share a genetic similarity, they must be part of some sort of transmission event. I was just sitting here thinking, if I share some genetics with Mark Westman here, does that mean we live in the same suburb? There seems to be some sort of error in inference happening here. Do you think that hasn't been examined enough because it's such a new technique that everyone makes this assumption because these two organisms are the same, they must be part of the same epidemiology? Or is it just a random event? Or is it that we only take samples from certain places, we never have a control? Like if we then look at Jackie also shares the same genes, but she doesn't live in the same suburb. That sort of error that's going on with these approaches.
Yes, no, and that's a very good question. In general, it's unlikely to find clonals if they are not genetically related somehow. It's unlikely, not impossible. So you always have to interpret the data with clinical data, with epidemiological data. Otherwise, it's very hard to infer transmission or to conclude anything. But for example, in tracing food outbreaks of listeria in raw milk, they go and sample of the range of bacteria that they have from public databases or from other cases of food poisoning in the area. And mostly, they first find the genetic link and then the epidemiological link through that. So it can help infer transmission through that. So yeah, it's not impossible.
Okay, I'll put you on the spot then. So during COVID, do you think the conclusions were valid every time there was some sort of genetic matching of the SARS-CoV-2 virus in patients, that therefore there must have been a link, there must have been some sort of transmission going on, even if there was no evidence of that?
So with SARS-CoV-2, I think it's a little bit different because it's a coronavirus that just started diverging 2 years ago. So you might find a lot of clonals there that might or might not be related. It's not the divergence that you have in other species that have been evolving for many years, and you have such a wide genetic diversity. So yeah, it might be, and also there's convergence evolution in which you think that some of your genetics tends to be likely because of an environmental pressure. So yeah, I think it's a different thing with SARS-CoV-2.
We relied much more on co-location being in the same suburb, as I was saying, because those divergence weren't there.
Hi, Barbara. Just to build on Michael's question and on a thing that you touched on in the lecture, in that metagenomics and metatranscriptomics is only available for first world countries. And just to kind of touch on what happens when we don't know, and if we go to a third world country and start seeing what might we find?
Yeah, so you start discovering that the genetic diversity of microbes is much wider than we rely on by having these studies in developed countries. So yeah, that's basically to highlight that the efforts needs to be towards developed and developing countries to have our full catalog of the whole diversity of the microbes, or at least the ones that are more important to us. And also in terms of the viral metagenomics, I know that there's a lot of international efforts to go to places that could be hotspot for the next pandemic to go and do metagenomics to try to study what could be the next pandemic, which is imminent.
Jenny Ann.
Thank you very much, Barbara. It's a pleasure to have you here delivering the address today. Just at the end, you mentioned the current conversation within Australia about a Centre for Disease Control. And I think there are a number of us that are wondering, how do we influence the formation of a CDC that will have a very strong One Health, focus? What would be your thoughts around how we as a profession and as One Health advocates can speak to or influence that process if we do, as a nation, move forward to establish a CDC?
Yeah, that's a very interesting question, and it's really how the groups that are creating the CDC see the importance of having a One Health approach and make it accessible, and hear the views of veterinary schools around the country, because I think that the One Health concept has been slowly pushed forward to a priority in terms of, for example, antimicrobial resistance, and now to understand the zoonosis and potential pandemics that could emerge in different places. But it really depends on the people that are creating it to make it accessible and to listen to all the points of view. The environmental of us, the ecologists, are also a very important piece of that puzzle, that big puzzle that has to be constructed. But yes, it's having people that are creating this understanding that the multidisciplinary approach is a necessity rather than an ideal of bringing people together. But yeah, hopefully they do. I saw a couple of days before, I looked for it on the website, so to see if there were any news. And there's actually a website that says that they are starting to create that. I didn't see anything veterinary mentioned there, unfortunately.
I was going to ask that same question, Jenny, and probably less politely than you did. How do we shift One Health away from one's health, which is often what it became very much around COVID. It was all about health. To be part of the conversation, don't we, Barbara, as veterinarians? I've got one more question for you, and it's maybe some of the things we were talking about before you spoke, but how do we use some of these advances to build in some of the surveillance systems that we might have on a an ordinary level, on a day-to-day or a month-to-month level, in the same way as we do for some of the things in human medicine around screening?
I don't know. I think that would be a slow process of incorporating a new technology that at this moment is expensive in diagnostic lab. Probably, I don't know, when I was working in Chile, real-time PCR machines were very expensive. They were nowhere to be found. And now it's something that's routinely used in veterinary diagnostic labs. So I see this as a natural progression of things lower the cost and are made more accessible to the diagnostic lab, in which they will start to incorporate it as surveillance or as a diagnostic tool. But at the moment, it's still a little bit costly and a little bit complicated to do all the data, the after data analysis. No?
Yeah, the processing.
The process.
When, it wasn't that long ago where whole genome sequencing was $1,000 now.
Yes, yes, exactly.
There are no more questions. Can you please join me in thanking Barbara for her? There is a token of appreciation. It's not wine, as you might imagine. It's from Aveda.
Thank you very much.
So, we've got drinks and food awaiting everyone outside, and conversation and real face-to-face human interaction. So, please join us outside. Thank you.